some
love
sort
that
of
stuff.
jelly.
They're
They do
growing.
make
Down.
pH sensors for ROVs, but they're very finicky.
Yeah, they are.
Let's see. Need to be calibrated frequently, like every dive.
Room for improvement.
Yes, no, it's definitely an area of huge interest.
Whoever's asking that question, you know, it's a what problem to tackle.
Even the common ones that are fairly inexpensive, but expensive for gardening and fish tanks and stuff like that are difficult to keep accurate. At least the ones that I've played with. There must be a laboratory grade one that's more reliable, or are they finicky as well?
Yeah, I think we had one, we used one in 2016
It's the red thing on
off
rock
of
bottom
Santa
right.
Barbara Basin.
Just
It looked like a shrimp to me.
Down a little bit. Look like a shrimp.
Yeah, we can make sure, but it had a shrimp shape.
Just directly below.
Yeah, just right below the screen.
Your Zeus right now. There it is.
Yeah, right there.
That's different than what I've seen before. Looks like it's basking on the rock.
So Brendan, that big rock that we saw,
Zoom out.
Center right.
Oh, I see it now.
oh wait,
Zoom in there once,
look
yeah.
at him.
Yep. Looking shrimpy.
Yeah.
Yes, this shrimp is in fact
Its
shrimpy.
legs are very long.
Nematocarcinus, something.
He doesn't move around too fast, does he?
I'll write it in
It's
there.
got like blue particulates on his antenna.
I'll write the name in the chat. It's cool.
So Brendan,
Yeah.
going
Yeah.
back,
Very.
oh
yeah.
sorry Steve, continue.
No, it's just a lot of marine snow stuck to it. Stuck to
Yeah.
the.
Are the shrimps filter feeders?
These, no, they're probably detritivores, so they're eating stuff on the bottom.
Ah.
Yeah.
So this guy's eating well. He's not struggling.
Presumably, but it's hard to
basking in
tell.
it.
Like, you know, we kind of equate, you know, particle size with particle quality, and that's not necessarily true at these depths. You know, these particles could just be masses of, you know, really low quality food, and might not provide, you know, sufficient sustenance.
So down here, it's more of quantity over quality.
Yeah, which is typically why you'll see,
Roger, okay, moving up.
which is typically why you'll see
Good, hey?
things with, you know, like better flow conditions. So they'll be on the tops of rocks or on the sides of rocks where you might have faster food transport. So it's tough for them to you know, be very selective on food quality, because you don't know when your next meal is going to necessarily come along. It's almost chance, but you can increase your probability of getting food of any quality by being in a better flow regime. There's nothing loose except all these fine grained stuff.
So Brendan, going back to that big, large rock area, someone in the chat wants to know, how do you think such a large rock is sitting with all these smaller pieces?
My guess would be it fell from higher up.
Just crash landed.
I don't know how long ago, or don't know what event caused it. Could be tectonic, could be something else. Could just be wherever it was unstable.
Quick zoom
And
on this guy.
Gravity did its job, but can't know for sure. It didn't look like a separate like lava flow, if that makes sense. It looked unconnected, disconnected.
Okay.
Sponge.
Similar sponge, yeah. Do you scratch up the bottom a lot, Dan, driving through?
What's that?
Does the bottom of Herc get scratched up a lot?
Yeah. It's covered in a stainless steel wire mesh, but if you look at it when we launch and recover, it's basically a skid plate.
How often do you have to replace that mesh?
As far as I know, it's been on there for probably 5 years.
So not too often.
That was terrifying.
It's pretty heavy
Sorry.
stainless steel mesh.
Jeez.
It also has skids, Delrin skids on both the feet, and we replace those once in a while. I think that the front gets worn out typically more than the rest of it, so we always land in the front of the vehicle.
So what would you say is the most durable or fragile part of Herc?
What's that center screen there? Orange and
Oh yeah,
red.
I see it. Sea cucumber?
Oh yeah,
Oh yeah.
that middle screen.
Looks like a sea cockroach.
Those would be horseshoe crabs, which we will not see, but very cool animals.
Zoom in there if you want.
It's kind of the color of the
What
thing I
is
was seeing
that?
before.
Oh wow,
Is that
you see all the gut contents of it.
Wow.
a sea pig?
That looks like a different species of sea cucumber that other than the ones we've been seeing so far.
That's bizarre.
Yeah, I would expect to
Liz!
see different ones down here.
Liz, wake up!
Wow, I think
That's really cool.
Yeah.
it's worth getting.
Yeah, we can try to collect it. I think Liz wanted more shallower species.
Yeah.
That's what I heard as well. Yeah, let's pass on
Yeah.
this one.
The gut contents are
But still, yeah,
visible, though. It's
good
cool.
observation
Yeah.
for identification purposes.
Good eye.
There's so much of it, too. Yeah, I think
Yeah.
this is a Cynalactid in that family.
So the back,
So clear.
the back of that area, is that an intestinal tract or is that something different?
Yeah, that's intestinal.
Huh.
I don't think this is one of the swimming ones. Yeah, it doesn't seem to be adapted for that. And this one seems to have these small little legs,
Yeah.
which are basically tube feet or modified tube feet. All right, all set.
Moving
Let's move on.
That's really cool.
Yeah,
Beautiful.
good eye.
Thank you. After I saw that other red one, I've been like just hunting for their maroon jelly.
Hunting for the maroon jellies.
Yeah. Not like that color, you know.
I'm
If we
surprised
see-
we actually saw one at this depth.
I guess
If
the
we see
third
another
was-
one of the shrimps, we're going to try a slurp collection
Oh, cool.
on the long-legged shrimp in our NOAA Animal ID guide. It's listed as Nematocarcinus species, not identified. So worth
Nautilus
a
Live
collection.
viewers, you may be in the process of seeing and witnessing the slurp of a new species.
My thruster's having a hard time getting me anymore to port here. It's just full bore.
Yeah, it'll sometimes help if you click
Cycle
on
the
and off
auto.
the auto head, but yeah,
Let's try that.
I'll come back a little closer there, got off track.
That's a nice big rock.
There's a shrimp there at center screen.
Oh, yep, there he is.
Is that,
Bottom left now.
is that one
wow.
of the benthic
Yep.
ones?
There it is.
Looks like he's
it's
swimming.
different,
I think
yeah.
it's a.
Oh.
I think it's a
Oh.
different.
Yeah.
Yeah.
This
Yeah,
is one of the
would never
swimming
catch him.
ones.
Here's a question for
I
Steve.
make a note in the book that we need to re-glue this joystick.
Yep.
It's annoyingly.
What is the shallowest target area that you're going to be on, that you're going, sorry, what is the shallowest target area that, what is the shallowest target area you're going to be during this cruise? That took way too long to say.
Probably, well, we're going to try and get as shallow as we can. We'll, might be doing a dive at Johnston Atoll sometime later in the, to be determined based on the weather, most likely. I won't give it
Oh, another
exact
sea
date and time,
cucumber.
but
There's another sea cucumber there.
at that
I
site
see.
we'll probably be able to get the
And
shallowest,
there.
maybe a few 100 meters.
Oh, wow.
That'll be fun.
See any shrimp?
Yeah.
Not for me, but
Well,
for you
I
guys.
mean, there's interesting geology there too. It's not
Yeah.
basalt.
Yeah, limestone.
I'm one of my favorite, habitats are these karst terrains
Oh, that's true.
we find. Yeah,
Is that where the caves potentially are?
yeah.
That would be very cool. Yeah. Karst topography is very cool.
So I want to fly Herc into a cave.
Grab us a speleothem or stalactite.
Any idea?
Could be a one-way trip.
I won't be following you, that's for sure.
Another question for the team. Any idea what the flucky particulate in the water column is? Maybe seep mat stuff? I believe it's all detritus. So like dead phytoplankton.
Can we maybe grab this rock?
There's
Is
a-
it disconnected?
Oh, look,
fecal
another
matter
shrimp
from
shrimp
up above,
bridge now.
Where does he see?
A little
Right
lower.
here.
There you go.
and it just kind of collects and then seeps
tell
down
if
to
it's
the
on
Hold
ocean
there.
floor.
position here, please.
So,
It does
shrimp
look
there.
connected.
Nice
Oh,
tiny
the shrimp
net.
as well, actually right next to it.
Yeah. Is it that long-legged shrimp?
Looks benthic, maybe.
Yeah.
Zoom in there.
Did you just gain your shields back in
Oh,
Halo?
yeah, there you
I
go.
did. I did.
Yeah.
Oh,
It's
yeah.
fine.
Oh, yeah.
Well, we can pass on the rock, but the shrimp is, yeah, cool.
Is that one of the ones you want? Is it?
The shrimp or the rock?
Both.
The shrimp, yes.
No on the rock.
Right. What jar are we on there?
Let's see. We have 2, 3, and 4 available.
2, 3, and 4. Roger.
It's got that blue
Can you
shimmer
move the
again.
jar to-
Oh, only at certain angles, too.
There's 4. 4 is ready for you.
Can you go wide for
Yeah,
me
yeah.
a bit? Not sure where I am in the.
These ones aren't fast at all.
You'll have to sneak up behind it. Yeah. That's probably the best way. It'll probably shoot backwards.
Oh, interesting.
Definitely shoot backwards.
I'm going to turn up the suction early.
No, the water, they'll sense the water moving, so we got to kind of time it.
All right. Just holler at me.
If I
Just
don't
watch
scare
out.
him away, trying to get this
So
thing out of here.
usually come up behind it with no suction and then 100%.
Yep.
All right. You got it.
Give it the beans.
Give it the beans.
Give it the beans.
So this is going to be the genus that is, assuming we get it in the chapter.
Assuming we get it. Okay, can zoom in a bit there.
Yep.
Tell me when.
Okay, go for it. 100%.
100%.
See, as soon as you turn it on, he sensed it moving and said, I'm out of here. They're really hard to get. Okay, turn it off, see if he lands again.
Well.
It's off.
So
That's a good try.
It's okay.
You were looking at a rock here, were you?
Yeah, but I don't want it anymore now that we're closer.
Starting to creep away from you up and above here, Dan.
What's that?
I'm creeping above and away from you. Let's swing around.
We're gonna move on.
You don't want that rock now that there was a shrimp on it. Gross.
Yeah, might have biology, DNA and stuff all over it.
Should have waited
Cooties.
a little longer, maybe.
Shrimp
before we,
cooties.
Before we tried it.
Oh, what about this rock actually?
I need to
Okay.
move on here. We're
All
getting
right.
run over. Sorry. I don't want to get too far behind Argus on this steep hill. I mean, Atalanta.
The ship is stationary now, so once we get Herc out in front, we might have an opportunity to grab a rock again.
Okay.
The blue glow of Herc kind of reminds me of something out of Tron.
Tron.
Which just looks very sci-fi.
Small bioturbation mounds, maybe, here. It's very unusual.
What were those, Steve?
Possibly bioturbated mounds. When you get sometimes
Quick-
animals that
quick
live
zoom
in
on
the
one if you want.
seafloor, they'll create small mounds. Could also just be a strange accumulation of rocks, but
Okay. Zoom out.
usually when the sediment is lighter in color than that around it, it usually means it came from a bit deeper.
Oh,
And some of these patterns are
yeah.
seem unnatural.
They do. Look at the Atalanta screen here.
Yeah, yeah. So those could be bioturbation mounds. You know, when an animal burrows in, they will often create these. Yeah, that's got to be.
Cool.
It's got to be biological.
I
I
have no
think
idea what animal.
that's a 3 for our lottery. Yeah, right.
Ticket.
Lottery tickets.
Or an
yeah.
M,
So
depending
we got
on which way
5,
you look
6,
at it.
3. All right, I'm writing this down.
I saw a
6,
2
3.
back there. So
And then the
Add it
Powerball
again if you want.
is several numbers longer than that.
It's okay, we still got another 2 weeks.
I'm seriously going to play this card when I get back home.
You know, if you win, the whole watch is
Okay.
on your card, right?
Exactly.
You got to divvy out some watch bucks. I'll mail you something, yeah.
Well, there's maybe a shrimp on your right side of Zeus.
Steve, you have the chance of winning dozens of dollars.
Keep moving here.
Yeah, another stocked
Alright.
sponge.
So, yeah, so only slightly more than a graduate student stipend. Ouch.
Too real, Ed, too real.
I'm gonna pull you off to the left here. This is a feature we're looking at.
There's
I
some
can get
kind of
there.
big rock coming up.
Yeah, looks like it. I'll probably have to go tail to tail here, maybe.
Yeah, we already are.
Roger.
Wow.
No life on this one, though.
Think it'll fit?
Oh, it's a one sponge. I think it'll fit. Oh, yeah, I see it. Nothing we haven't seen before, though.
Is that a loose rock at the base of the big one?
I can't tell.
Pull that one out, the whole thing's gonna fall.
Oh gosh.
That's how it gets stuck.
Okay, I'll come back towards the...
This.
We can start moving again if you want.
Bridge nav. Can we move 40 meters 200, please? Thank you.
Oh, getting Atalanta in the still cam.
Oh,
Oh yeah.
Ooh, ominous.
It's quite a struggle for this one thruster to pull the tether back around.
what's that?
It's a fight for the one thruster to pull the tether back behind itself.
Yeah, it is.
I've always been curious, if you could put a soundtrack behind all of this, like this kind of footage,
Almost
what
there.
soundtrack would you use? What song?
Dire Docks. I'm not going to elaborate at all on that.
Thanks.
We're all caught up, so if you see any rocks you like, holler. Not that they're
Understood.
very abundant, but
Everyone do themselves a favor and look up Dire Docks.
Nope. We're just gonna sit here in confusion.
Does that look loose right here?
Could be. Yeah, both of those are loose.
Oh yeah, there's multiple here. The other two above it look too thin.
You like the one at the bottom of the screen there, do you?
Yeah. Let's do it.
Brandon, did you hit us with a Mario
Pick
64?
up that rock. Yes.
Yeah,
Okay. All right.
of course.
Pick
Oh,
up
you
that rock
Yeah,
right there.
all right.
do you want to hold position?
You better be quick about it because you're going to get run over.
Oh, Subnautica soundtrack. Yeah,
It's
okay.
coming up.
I see you.
Bridge nav.
Some people knew it.
Some people know.
No,
Hell
you can
yeah.
keep moving.
Oh, cancel that. Sorry.
The deep. Okay.
All right, where are we going with this thing?
All right.
Pick it up first.
I think the Subnautica soundtrack could be a little too scary around here.
There's plenty of water levels to choose from.
Let's just do the shallow. Let's not go to the dark zone or whatever it's called. FF17. Okay.
Nice.
Okay,
Whoa.
I'll show it to the
Oh,
camera.
good
Not
rock.
too close, but
What do you think, Brandon? Good
yeah,
rock, bad
it looks
rock?
great. Looks awesome.
All right.
Somebody know our depth? Okay, thank you.
okay, I can put it in
All
the
right.
box. Go on one of the back ones, maybe.
Yeah, probably starboard side.
Yeah, C or D?
The
Or is it too big?
jaw lock button is probably not working, so you'll have to
Raj.
keep your finger on the trigger there.
Oh, sorry, push core.
It's a good Argus shot. You can
Which box,
snap.
sorry?
C or D?
Aye, all right.
Or you can also drop it in F, whichever one's easier.
I'd like to put it in F.
Okay.
Yeah, let's go for F.
All right. Well, the jaw lock button is working.
Of course, now it works. It's intermittent.
All right, so
Nice.
that's an F rock.
Nicely done.
Nice. Data, was that 097?
That is correct.
Thank you.
Can I lock it here?
Yeah, you can kill it there.
That was fun.
Nicely done.
Thank you.
Yeah, nice.
Nicely done.
Rock sampling on the fly.
Practice is paying off.
It is.
Bridge nav.
So proud.
Thank you, Matt.
Same step, please. 40200. Thank you.
And for our viewers at home, if you want to get just a sense of how barren this kind of place is, if you switch on over to channel 2, you can see our Atalanta view of Argus and this sea of just rock fragments.
Here's a boulder.
Ooh. Looks like it has a sponge stuck on it.
Or a barnacle, yeah. I'm not sure what at this distance. Another, actually, can we, down in the middle here, there's a red something? Is that a shrimp?
Yeah.
Or,
All right.
not sure. I think it's a shrimp.
Zoom in there.
Yep.
Oh, yeah.
It's the same one, right?
You want to try again?
Sure.
Let's do it.
This might be easier angle, huh?
Yeah, it's a little bit bigger.
I didn't realize they'd get spider-like like that.
Okay, kick
Yeah,
away.
it's this one family is long-legged.
Reminds me of a daddy long leg.
For those of you
Although those aren't
arachnophobes
spiders.
watching, we're so sorry.
Sorry, daddy long legs aren't spiders.
Stop right
Really?
up behind.
Yeah, they're not arachnids.
Really?
Nope.
You want to stop moving?
Are they
Wow.
insects?
Yeah.
Bridge
They have
nav.
6 legs.
Oh.
Can we hold position here, please? Thank you.
You want to show me the jar cam there?
Yep.
And 4 is ready for you.
Roger.
Oh, this is a great shot on here. He's prancing.
Okay, Dan, zoom in there for us.
Look at that shadow.
Dun,
Ominous.
dun, 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 dun.
Oh, man.
I think he knows.
Eat 'em.
What we need is to set up the Magnum, the ISE, distract him, and then come in from behind. Set a trap.
Guerrilla tactics.
Blind with the porch lights.
I think you just need to install like a harpoon on Herc.
A mini harpoon.
They're
Giant
actually-
butterfly net.
They're easier to get with the net.
Oh, yeah, that
Oh.
makes sense.
The next upgrade is the harpoon gun.
Hard to do.
Do we have that bucket scoop net thing?
Can you harpoon a shrimp?
Probably not very well.
Okay, zoom in there, good place.
Yeah.
Okay, suction up.
Suction on. Oh, so close.
Oh,
Wow.
oh wow.
Almost.
He was
Good
starting to
try.
come towards it too.
Almost.
Yeah, I
Cheeky buggers.
Nice try.
choked right there
They're really
No.
fast.
at the end.
They're tough. This is why a lot of mobile animals are unidentified from the deep ocean.
They're evasive.
Speaking of mobile animals,
Can you show me the
someone in the chat
the
says,
five on
how
the-
well do the shrimp handle the pressure change when coming to the
5 on the
surface?
graph on the bubble cam?
As far as we know, they're not substantially affected, but the temperature is a bigger issue for them.
Really?
You know, the temperature down here right now is
All right,
just a second.
I can put that camera, 2 cameras
1.4 degrees C, so
Bridge nav.
also known as a fine New England day.
Can we move 40 meters 200, please? Thank you.
All right.
Or I don't know, 1.4. That's maybe it's chilly. It's just above freezing.
It's almost as cold as this van.
Yeah, so they're going. They're
Almost.
Sorry, Steve.
You didn't know the old control vans. This is tropical.
This is luxury.
This is luxury.
Luxury.
It's like a hot tub.
Yeah, the old control van had a temperature gradient that was I don't know,
Severe.
infamous. Yeah. One person would be dressed up in a winter parka, and one person would be sweating.
Oh, really? From seat to seat.
Yep. The science row was always comfortable though, which was important.
Ooh, a brittle star.
If you were sitting
yeah.
next to the racks, it was another story.
A little chilly.
There was one year where the AC gave out. This was a long time ago, and we had big, like, industrial, garage fans blowing in here just to circulate the air. Couldn't hear anyone if you talked to them side to side, so the only way to communicate was through the headset.
Yeah.
So it sounded like a hurricane.
Still pretty loud in here.
And then another question for the ROV people. How long does it take for Herc to descend all the way down there?
Goes down at 30 meters a minute and comes up at 25.
What was your estimate
15,
limit?
sorry.
So 30 meters a
15 to
minute.
20 on the way up.
Let's do the math.
1
I
knot,
think our nav has an estimate of ascent time.
2 knots down,
Is
1
this
knot
a
up.
bamboo?
So 1 knot
Looks
per
like
mile.
it. Yeah.
Oh,
Let me
cool.
take a peek if you have time.
What do you want? Just a shot of it?
Yeah, just a shot of it.
Go
Yeah,
ahead.
right
Zoom
now
in there,
we are at about 3,500 meters. If we were to come up right now from this depth, it would take about 4 hours.
Yay.
Can't see its nodes very well.
There's one.
Come around the side.
It's amazing how all the
Yeah.
polyps are facing one direction.
This one might be calcified over.
Okay.
I see.
Jeez. So they face
Yeah, it
down
is very
current,
tough.
right?
Sometimes the nodes are very difficult to see. So this is probably a bamboo coral, but yeah, based on the density of the tissue, sometimes it's really hard to see the nodes. Can we walk up the colony?
Sure. You can
I
zoom
believe
in a
this
little
is
bit.
Bathygorgia or something very similar to it.
Thank you to the viewers' support from their Discord server. We will slurp
But
straight.
yeah, these are exceptionally either difficult to see or very long
I'm going to turn
internodes.
auto off. You're going to get a tug here.
Roger.
Oh, there's one. Yeah, I think they're just very, very long nodes or internodes, which is
It's max.
something a little bit unusual. This one looks like it might have captured some sort of particle.
the
Ooh.
polyps. All right.
So how do the polyps capture particles to feed?
Yep. Yeah, and then they'll usually close up.
Like little hands.
Yep.
Neat.
Yep.
That's so cool. It's like a centipede times 1,000.
Yeah, this one is very large for this depth. Must have be really kind of stable, long-lived
Go in.
animal down here.
Okay, I gotta go get pulled here.
Is this our first coral of the dive?
No, they collected one a bit deeper,
Oh, okay.
Primnoid.
Okay, I want to swing around to port.
All right,
I think we've seen maybe
switch over here.
half a dozen corals or so on this watch. There's an unusual shape down here. We pivot down below this rock.
Yeah, I
Just
can't.
wanted to check something out.
It looks like a
Something
sea
red there,
cucumber on top.
Snap zoom right here at the base of this
Oh
boulder.
yeah, it's that sea cucumber.
Yeah.
That type of sea cucumber from earlier.
I was looking at this shape
The
here. I
blue
think it's a
thing.
fish.
Yeah,
Oh,
it is a fish.
Yeah, good
Oh,
catch.
wow.
Oh,
There,
I
quick
don't
zoom
see it.
there.
My,
Dang.
oh,
Good
wow.
eye.
Oh, wow.
Whoa.
Is that a cuskeel?
Cause that's where the brain and the mouth are moving at the same time.
Kind of looks like a grenade.
Yeah.
Your thought is being composed as you're talking.
Okay.
Yeah, I don't know. I think it looks like a cuskeel. It's an unusually active one for this depth, so moving around quite a bit.
So ones at this depth, they usually are like kind of
Yeah, they're usually slow.
still?
Yeah.
Ah.
Although, you know, we are in a very different area than we were yesterday. We moved about 18 hours to the northwest. So, you know, productivity might be a slightly different over here, but I don't think it would be that sharp of a gradient. You know, I think
I'm gonna try
that this
coming around
productivity is probably representative of this side of the atoll. So if we continue to dive over here, we might see more higher density communities. Not at this depth, because this site only goes a few hundred more meters shallower. But we're going to be operating over here on the west side for the next week or two. I don't even know how many days are left. A while. I don't think we're halfway through yet.
Yeah.
Looks
Speaking of
rat
corals,
tailish.
Our friend Jason asks, did you examine the gall on
How's
the
that
coral
color?
sample from yesterday?
You know what? I did not yet. I was kind of focused on the predation experiment yesterday in the lab, so I didn't get a chance to look at the gull from the primnoid, but I will take a look at that. There was also a gull on the bamboo coral we collected. I'm not sure what that was. It could have been an anemone, but it was a very unusual shaped gull. So all of these things will probably have to be dissected back in the labs on shore, where it's a little bit more stable and we can have a little bit more precision. We're still rocking around a fair bit out here, and doing some very, very sensitive dissections is not always easy or possible.
Bridge nav. Same step, please.
How do
40200.
you dissect a coral?
Thank you.
It's not really, so if I was trying to get tissue out of the coral, I would just scrape off the polyps
Oh,
off the branch. But if I was looking at a gull, I might want to, you know, look at it under the scope while I'm doing it.
oh, okay.
It's not, yeah, it's not very, you know, fancy work. It's not like sea cucumbers, where you kind of see what you're doing before you even get in there. Just slice it open, take out the gut, for example.
I
For
see.
corals, it's more simplistic.
What type of coral is the most interesting to dissect?
Interesting. Probably mushroom corals are pretty interesting. They have very, very dense anatomy.
Oh, that's quite the
They look very
overhang.
soft and squishy though.
Yeah, they are.
Nice.
They pretty much have the consistency for what they're described for. Maybe a little bit more dense than a mushroom, but
Ooh.
Is that just detritus, or is that a white thing that's different floating in the bottom middle?
It's a swimming worm.
yeah, maybe a polychaete worm. Yeah,
Ooh.
you can see it undulating.
I have a coral digestion question. I
Large.
guess I have all the bathroom questions. The alvin bathroom question. Now I have coral bathroom question. So the individual polyps, they kind of have these little fingers, which I assume catches food out of the water column and pulls it toward a mouth in the center of the polyp. Is that correct?
That's right.
And
Yep.
then what happens?
So then they,
Cucumber.
oh, cucumber. Then they kind of relax the this oral disc they have on top of the polyp. They'll relax that and they'll open up a sphincter into a gastrovascular cavity. Basically, it's a two-way digestive tract. Pretty much, the food particles go in, they close that sphincter, and then they'll digest whatever meal they took, and then whatever they can't digest, they'll spit back out their mouth.
Okay, thank you.
And then the nutrients will be distributed through the body, through the colony.
Good question.
Do the nutrients that one polyp takes up, does it feed only that polyp, or does it transfer somehow, like through the stalk to others?
They're shared, yeah.
Okay.
The food is shared.
Cool.
Can we zoom, is this one of those tunicates, this quick snap?
Sure.
Yeah, that's what it looks like. Yeah, tunicate. Okay, thanks. Something rolling around on deck out there. Is anyone else hear that?
Yeah, it's loud.
Ah. Question from chat. I'm not sure entirely how to word this, but is there a fungal equivalent to the deep sea? What happens to deceased creatures that aren't eaten?
Can you repeat that question? I didn't hear a couple of words.
I'm not quite sure how to word this, but is there a fungal equivalent to the fungal equivalent of the deep sea? What happens to deceased creatures that aren't eaten?
Yeah, there are fungi in the deep sea. We usually list them as microbes. They're very small. We're not going to find like
Another
large
shrimp,
reproductive structures like mushrooms or things. They're mostly small fungi, microbial sized, but they will break down material as well as bacteria, lots of other animals, viruses and things, all part of the microbial ecosystem. But any large tissue is probably going to be eaten or broken down by larger detritivores. They're called animals that eat, dead and decaying, chunks of things. Maybe even smaller particles. But yeah, there are all those same processes at the surface going on down here in the deep sea.
Really?
Yep, microbial degradation. Yep, fungi, the whole deal.
There you go.
It's just missing photosynthesis.
Yes, no photosynthesis.
Minus that part.
Yep.
So then, here's another question. Somebody asked, are there are there leeches or a
Zoom in
type
there real
of
quick,
leech
Dan.
that would live at the bottom of the ocean?
Yep, there are probably leeches. I have never seen one myself,
Oh, look at that.
but as far as I know, the group that leeches
Wild.
belong
Wide,
to,
yeah.
the Hyrudinians, I think they are present in almost every marine environment, I believe. So I think I would have to consult a specialist on that, but
You got a rock.
I believe they are present in the deep ocean and on the seafloor.
Big rock, right? Oh, you got it there.
And then really quickly that question, are you guys observing this in 4K quality or HD? We are observing this in 4K, the same kind of video quality that you can view on our Nautilus Live.
Oh, fish.
Ooh,
Ooh, fish.
I think it's HD.
another cuskeel?
Skill.
Oh, that's a long pectoral fin. Oh,
That is so cool. Wow.
wow.
It's pretty chill. Jeez.
Oh.
Nice shot.
Nav, I
Still
have
fun.
a update request for direction change.
Okay.
Can we head off to the northwest towards the area of steeper slope, kind of lateraling along the depth we're at right now?
Sure.
Maybe
Can you hold the
200
boat
meters
for a
or
minute?
so.
Okay.
And Steve, another question from a viewer. So you said that the same kind of the same kind of systems on land can be mimicked or the same in the ocean. Does that mean, like someone says, could a barren seabed be used to cultivate deep sea organisms, like a deep sea garden?
That would be challenging because I think a lot of deep sea animals are very finely tuned to their environment. Kind of artificially creating an environment for them is not always going to work or be possible. And things like that are very slow to happen in the deep ocean.
Hold on a minute. It's we got a steep pit here in front of us.
So even if you did do
Which
something
way
like
are you
that,
gonna
it's not
plan on
even
going
known
now?
if you would be able to tell if it was a success or not for, years, decades, centuries.
What's going to be your new bearing?
Thinking 290 to start out.
Rather. Think about that for a minute.
You ready?
No.
No. Okay.
We see some questions in the chat about the history of Johnson Atoll. If you are interested in the history of Johnson Atoll and the effects that it's had on the sea environment around us, you are welcome to go to our website where if you click on the More Information tab, you can find a bunch of guides and a bunch of information about Johnson
We're,
Atoll. We also thoroughly
right
encourage you to look up your own
Atlanta at the moment,
sort of
and
information and find your own
it's,
opinions and other pieces of
we've
information about Johnson Atoll
come
on
from
your
the
own time.
flat off to a steep cliff here. So.
I also think Yvonne may know quite a bit of history. I think he may be on the
Come
next
up,
watch.
That's true. So if you're
come
willing
up,
to stick
Tom
around
Eggers.
for another few hours, you can also ask your questions then. I'm sure he'll be able to answer it a lot better than any of us can.
Yeah, so I mean, if we could head,
Okay.
kind of in this direction that that you're planning, I don't know, maybe we'll go 200-300 meters or so just to see what it looks like over there, and then
Roger
could
that.
not-
We'll
you can
resume
move it
our,
now.
our
You ready?
normal
Yeah, Roger.
course.
Bridge nav.
Bring your head around to the west there for
Can
me.
we move 40 meters bearing 290, please? Thank you.
So we're going to go along the slope
Yeah,
now.
yeah, we're going to go along
Gibson?
a little bit.
So we
Oh,
end up pay more attention to the 20-meter range ring there.
Yeah, roger.
So I can't.
Currently heading Atlanta 270.
Yeah, Raj, I'm going to try and stay on the upside, but it's fairly steep here all of a sudden, so it could sneak up on us when we're
Roger.
creeping along. Depending on how it, if we're going along like this and the ridge comes out, it'll.
Understood.
oh, that's right. Going back to the leech question, Tina was just reminding me about fish parasites. Also is where we see a lot of weird animals. Fishes often will have isopods and parasitic barnacles and things like that. Copepods attached to their gills or sometimes inside them. Leeches would be common on the outsides as parasites of fishes.
What are some of the more common parasites that we see around here?
There might be another fish in the top left of the screen.
Probably isopods and copepods.
Keep going left. It's on the DSC, on the...
Oh, actually, I have another request. On the top of this sponge here, there's a coral that looks like it's stuck, like a red. Or maybe it's a brittle star, I don't know, something red on the top of that sponge.
Running low
Push
on,
in there a bit.
running low
Oh,
on
it looks
tether.
like a crinoid.
Crinoid there, yeah,
Come
and
down
then
a
a
little bit.
brittle star. Okay. I thought it was a stoloniferan for a second.
It's cool.
Ooh, what's
Never
that?
mind. Caulophacus, yeah, with
Okay,
brittle
cool.
star and crinoid.
Here's a question regarding the detritus all around us. Any idea why the detritus on this site looks a lot bigger than the previous dive?
Yeah, so we are a fair ways away from our last site, about 200 kilometers or so. So at this site, we might be in a slightly different productivity environment, slightly further north and west. Also, we're deeper, and sometimes when you go deeper, the material in the water column becomes more flocculent, so that means it sticks together, forming bigger clusters the deeper you go. There's also a possibility that, to check on this with our data set, but that we could be sitting on some sort of density layer that would result in kind of slower sinking or settling velocities of particulate matter, which would cause the material to clump or stick together with a greater frequency, greater probability. Those are all possible scenarios.
You okay to keep moving in this direction?
Yeah.
Bridge nav.
So then, Steve, what are the sort of kind of organisms that we've seen?
Same step,
Around here.
please. 40290. Thank you.
A lot of what we're seeing here are tunicates and sponges.
On this dive. I've seen a few corals, but tunicates and sponges, more common.
So this viewer specifically is very excited about jelly, saying, "Hi team, are you all hoping to see a nomad jelly? What is some of the maximum size that these specific jellyfish can grow to?"
I don't know what that is. Let me look it up. Sounds cool. Yeah, I don't think we've seen anything like that, and I don't know if we find them this deep. So I wouldn't expect to see something like that here.
Sorry, viewer.
I'd be excited if we did see it.
Sorry, Steve, did you say we don't expect to find any jellyfish this deep, or just that particular species?
That particular one, yeah. Can we zoom here?
Sure.
Is that, I wonder if that's debris or if it's something stalked alive.
Push in a bit there.
Sure. There, yeah, just sponge rubble. All set, thanks.
Hey Steve, that fan behind you on the AC, what's it set to? A little black box on the wall behind
Yeah,
you.
it's off right now.
Is it?
Yep. You want me to turn it on?
No, I'm just curious if you'll have cool breeze that we don't usually have in here.
Here's another question. If sunlight managed to reach the deep ocean, would you think there'd be more life? Could you set up a full spectrum LED lamp at the depth? Never mind. If sunlight managed to reach the deep ocean, do you think there'd be more light? Or more life?
So the way I would answer that is kind of in terms of how deep light penetrates, which is variable. Maybe in the higher latitudes where there's more stuff in the water column, we might see a photic zone of like
Can I give you a DVL reset?
Sure.
60 feet. So not very deep at all, and then it gets kind of dark or beyond maybe 50 or 60 meters, you won't have any light anymore.
What's it for tether?
But here it's not unusual to get light down to 175 to maybe even 200 meters, I think. It all depends on the clarity of the water. And yeah, you can end up with different species at different depths. But I don't know if there's a direct correlation between amount of light and biodiversity or abundance. You might just see different species.
And then, really quick, I'm going to answer that question in chat because I know this answer. What is the deepest this vehicle can go? Well, Dave from Columbus, Ohio. This vehicle, specifically Hercules, can go to about 4,000 meters, and I believe Atalanta can go to about 6,000, Gibson?
I'm going to defer to Dan on that one.
Yeah,
Dan?
that's correct. Yeah.
That is correct. Hey, look at that. I remember my trivia. And then,
Happy to keep moving.
Sure.
as a follow-up
Bridge
question,
nav.
after Lynette does her thing,
Same step, please. 40290. Thank you.
how much math does one have to know to go into marine biology?
You do need to have a solid background with math for statistical purposes. If you want to do oceanography, probably more so. But we use a lot of statistics, especially because statistics help us, when we don't have enough data necessarily to collect,
every little piece of information we need for a study. We'll use statistics to try and sample and determine if we have a good enough sample of data to base our conclusions on. So that's something that would be important from a mathematics perspective. There's also a fair bit of chemistry, depending on what you do in marine biology. Chemistry also requires mathematics. So yeah, it is important. Do I use calculus on a daily basis? Probably not on a daily basis, but it's important to know for some aspects of oceanography that might be helpful for marine biology, certain parts of marine biology.
Zoom in on that guy if you want.
Starting gauge
Like if
checks.
you wanted to go into physical oceanography.
Yeah, very important.
So stay in math, kids.
Yeah, looks like another smaller Bathygorgia. These sponges, these sponges remind me of the wacky waving inflatable arm flailing tube man.
Okay.
Just 3 of them. Are we in, can we grab rocks?
Sure.
Okay. I will keep my eyes out.
Gauges look good. Craft comp is down to 3.
Got it. It's falling, is it?
Yep. Started at 4.
Yeah, a lot more outcrops now that we moved further west.
That's our delta. Come
Let's,
down a bit.
you don't have to write it down, but let's check it a little more often.
Roger.
So Rob came in here a bit ago and was saying maybe this steeper ridge, or a steeper part of the ridge we're headed to, might have more fresh rock faces. So that's one of the reasons we're heading over here, to see what the rock looks like on the steeper slopes.
These slopes really rock my world.
Gotta use a different word. You can
than
show
rock.
yourself out after that one.
Savage.
Only the highest quality rock puns on this watch.
This looks like a basalt.
Is that one okay? Let me zoom on the sea star coming up on the right here.
Take it.
Sure.
We'll give it a 3 out
Go
of 10.
ahead, Dan.
Sure. I just tried to use a different word, you know.
I only know rock.
All right, so this is
Oh.
a
Wow.
sea star in
Beautiful.
the family Solastaridae, which are sun stars. And it's a 6-armed sea star. I know we have collected this one on other occasions nearby. I don't think in Johnston Atoll. Actually, I might be wrong about that. We might actually have one from Johnston.
And then answering that question, as a follow-up, is it because how long the cord is, or is it because of the pressure? I believe it's just because of the pressure and how much pressure these vehicles can take.
Yeah, keep going.
Yeah.
Yeah, the foam and the
All right, we're
main
all set
electronics
once we get a
bottle.
good imagery of this.
are
Okay,
rated to
go wide.
4,000 meters
Bridge nav.
This similar looking star was collected
because of the pressure.
Can we move 40
Uh,
meters bearing 300, please?
in 2017
Thank you.
on Oceanus Explorer cruise. I think it was collected at 2560 meters or so at Wetmore Seamount. Although in this
And the hunting
photo
gauges look
it
good.
looked like it was a 5-armed star, but they still classified it as Solastaridae. So maybe it was missing an arm there, looks like. But it looks like identical sun star.
So Brendan, here's a question for you. In this environment, are there any geologic processes that erode the rocks here, akin to weathering and erosion in erosion processes that occur on Earth's surface or in shallow marine environments?
No. If you have strong enough currents, you can erode underwater rocks, but we don't have that here. There are some theories about salt erosion, which perhaps sort of similar to like aerial, I forgot the word now, but yeah. And then there's gravitational erosion, which you see, you know, just fractures or some kind of unstable part of the rock. Gravity eventually will take over because, you know, there's gravitational force at all times and cause the rocks to cause rock falls and stuff, but no like scouring erosion for the most part. So these environments will last a very long time.
Discoloration.
At least until subduction takes them over.
And then answering the question in chat, sorry if I missed it, but can y'all introduce yourself? If you'd like to know who's on watch, if you go to our website at nautiluslive.org, you can see all who's
Grab
on
this rock,
watch,
maybe. Yeah.
what our names are,
Go
how
for
we
it.
got here, so on and so forth.
We're going to pick up a rock right around here.
Maybe this.
Can
Right.
we get that one?
Grab that rock up.
Do you need a pause?
No, I think we're good.
Which rock, sorry? Steve, can you circle it?
I get the minute out.
Kind of round one in the middle.
Ooh, jumpy.
Turn your draw 90. You're on a steep hill there. Turn your draw 90, man.
Come on, 90.
There you go.
Ooh,
Nice one.
Nice.
thank you.
Good rock.
It's not very angular, but it is thick. Yeah, I think it's good enough to take.
Okay. Stow it.
We can double up on F, maybe?
Roger that.
It'll also probably fit in C or D if you.
Okay.
Either.
F will be easier for him to, you can try C or D, it's up to you.
This one's smaller than the previous one in F, so it should be easy to tell apart.
And to answer really quick that question in chat, any Dumbo octopus sightings on this expedition? I don't believe so, but we are
No.
keeping our eyes peeled for them. So stay tuned, and hopefully we can
Not
see
yet.
one.
Maybe
It's
maybe
okay
in the
to
next
double up
several
enough?
days.
Yes, I think when we're closer to Johnston Atoll, tune in then and we can see more of those types of animals.
Yeah, I think you can put it in F. Did it go in?
Yep.
Nice one.
Nice.
Speaking of animals, another question. What kinds of whales, if any, live near Johnston Atoll?
It's beyond my specialty.
I think that answers our question. We don't know.
You know, I think you probably would get sperm whales migrating around here, maybe humpbacks over the course of a year. I know that further north you do find sperm whales and humpbacks in the winter especially. So
Nicely done, Goose.
it's not impossible or unreasonable to expect that they might be around here.
To come up on
But in terms of if you're
Atlanta a bit when
hoping
you
for a
come
whale
down.
sighting, you might be
Ready
out of
for
luck.
another step?
Very maybe
Bridge
altered
nav.
rocky bits here, or
Same
oxidized or
step,
something.
please. 40300.
So what do we do with these rocks? Someone in the chat wants to know, after you run tests on them, do we just throw them overboard into the water?
I assume they want to know with like our collection
Yes.
repository stuff. Yeah, I'm not. We definitely put it in a repository. I always forget the name, but the rock repository is the Marine Geological Samples Lab in the University of Rhode Island Graduate School of Oceanography. Thank you. Yes, they go there. Some of our geologists might take some for further data analysis as well.
Will you be taking any specifically?
Atlanta heading 270.
Yes.
Roger.
Really?
Yes.
What kind?
What kind? Basalt. For age determinations, as well as potentially chemistry, geochemistry. The ones that look nice, basically. We've got a good amount of altered samples, but also a good amount of basalts, so there's some potential candidates for good age dating. But even if it is a basalt, you never know if the ages are going to be good. Because argon can be a ficky element. Yeah, because like...
Colder in here tonight, isn't it?
Yeah.
One of the, I don't know where one of the fans was turned up a little high.
Is it this, do you think?
What's that one set
I
to?
don't know.
Press
that points that way.
see the little button on the very left bottom? Yeah, press that.
A crack
that
in the
one's
rock,
only
that's
on
kind of
one.
cool.
What are your fans set to over there, Dan?
Cold.
There's 3, well, wait, it doesn't change the temperature, it's just the fan speed. So there's 3 temperature readouts in the rack in front of you. If you touch the bottom left button, a number will pop up between 1 and 6. See the 3 temperature readouts up there up above, in the far right-hand rack. Rack 4, about 2/3. Yeah, if you touch the bottom left button on there, there will be a number between 1 and 6. One rack up. And those little numbers. So there's 4 little buttons on there, the bottom left one with a little fan symbol on it. Really? You sure that one in the back is off? Feels like that one is.
Then to really quickly answer that question in chat, when you refer to chat, is there a platform with a live chat that you're streaming to, or is it just questions coming through the website? Well, V, it's for now, all of our questions from chat are just coming in through our website. And if any of you viewers are interested in it, you can go to nautiluslive.org and scroll down past the little live stream, and there's a bar where you can answer or ask any question. I will do my best to answer.
Do you know if, well, we'll pause for a second. Do you know if the other communicators call it chat?
I like calling it chat. I know that our other science communication fellow
just turn it off.
on the 4 to 8 watch, Megan also calls it chat. I'm just used to my Zoomer Twitch chat, so I'm just gonna call it chat.
Steve,
That's what I figured.
that's about 200 meters west-northwest. Do you want to
Okay.
continue in this direction or start toward waypoint 2?
Yeah, can we go another 100 meters or so?
Yep.
Yeah,
it's definitely that one.
if we see another big outcrop in that 100 meters, pick up another rock, I think.
Do you want to head more northwest, or is this bearing okay?
This bearing's okay.
Okay.
Yeah, we can go up the slope a little bit while
Okay.
lateraling. Yep.
All right. Yep. Didn't want to backtrack too much. So.
Yep. Yeah, we'll
Bridge
kind of do
now.
a zigzag Z shape. Heading back towards the southwest.
Same step, please. 40300.
Sponge.
Thank you.
Oh yeah, there's a sponge.
Hey Dan,
coming
what was the fan number on the right hand
up.
display up there?
1.
They're all 1?
No, not everyone. Left one is 2, and middle is 1.
All right. Could probably set the very left one to 1 as well.
Okay.
See what happens.
That's a very large polyopogon, I think.
Alright, well, yeah, I mean, whatever's of interest here, I think this is exactly what we're looking for. Alright, well then we'll have kind of southwest.
Yeah, that's better. Quieter and not sitting in a wind tunnel.
Yeah.
And then this is a question for the entire team. During these steep areas, has anyone seen a rock fall or a rock slide?
I haven't seen one, but this is my first expedition, so.
This one might be polyopogon.
That's fair.
All right, so yeah, we'll do another 100 meters, move from here, and then we're gonna just head to waypoint 3 after that.
Okay. Sounds good.
And then really quickly going to answer that question in chat. Why is chat disabled on the YouTube live feeds? So we get our chat from a different source that's straight off of our website, so it's easier for us to monitor. So if you want to ask questions in chat, just go to nautiluslive.org, and then you can ask our team chat questions, and we will do our best to answer it.
Go ahead, Dan.
Sure.
It's another bamboo coral.
Bamboo coral. Not sure why it's all closed up. Maybe it had just consumed a meal or something.
Is that just that the polyps are closed? And it has that
Yeah,
lumpy
yeah, the
appearance?
polyps are closed.
Oh,
Yeah.
interesting.
Yep.
There's another one sticking out.
Oh, yeah, you can see some of the sclerites arranged in the body wall of the polyp. They look like they're oriented kind of across the polyp, like perpendicular to the polyp.
Yeah.
Great shot.
Oh, wow.
Nice.
To
This is
answer that-
actually really useful. We can almost get it to clade using these shots. It's very, 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 very fleshy bamboo coral.
Sorry, what did you say you can do with these shots, Steve?
We can get at some of the diagnostic characteristics that we can only look at, you know, if it's on deck, for example.
Wow.
So this is helpful, like to see, you know, how the sclerites are arranged in the polyp, the shape of the polyp. All important for getting, you know, better than family level
Awesome.
identifications. Cool. Thank you. All set.
then to answer that question in chat from Karen, currently the information panel on the right in the stream view does not have the ROV depth. What you can do is you can scroll down and click that more information tab, and then an entirely different page will pop up with a bunch of more information as well as depth, oxygen saturation, a couple other things that's really interesting. So
It's
go ahead
holding
and
at
give
3.
go ahead, give that a look. So Brandon, here's another question for you. When you pick up a rock and you look
I'm
at
gonna
it and
give you a
say
minute to get out front.
not a very
No, you
angular
can move.
rock, but a good rock, why are angular and/or sharp rocks good rocks?
Bridge nav.
Because when you have like spherical
Same
rocks,
step, please. 40300.
a lot of organic
Thank you.
material will form spherical rocks because they'll be covered in crust and you can't really tell the difference, but shape is all we have to go off of, and basalt forms angularly. Angularly, is that a word?
Yeah, okay.
Forms angular vertices. So yeah, we want something more angular, but can't always get that, so as long as it's thick and the crust isn't all that there is, which can be the case when it's a thin rock, then I think it's okay. But I don't have much experience picking up rocks, so if you ask that again when Rob or Kevin are here, they might give you a better answer.
And another rock-based questions. Do crystals or geodes form in the bottom of the ocean?
Crystals, yeah, crystals, minerals, they form. We found very big plagioclase and clinopyroxene crystals. Somebody might be mad for me calling them crystals, but minerals. And then geodes, I want to say yes.
We did find one
Yeah.
earlier this year, yeah.
Okay.
Your lab mate, Amber, broke open a rock and found a geode from the Lion Islands. It's pretty cool.
Oh, that's cool.
Small, but you know, it's geode.
So what do you do with those geodes? Do you send them back to the lab? Can you keep them as souvenirs?
Well, we're in protected water, so there's no souvenir keeping. You know, all of this research that we're doing and exploration is permitted very specifically. So all the specimens are have an agreed storage location that is agreed upon by the monument and those who apply for the permit, and all those samples are going to go to the same MGSL lab.
And to answer that question again, what's your favorite thing about this job? My personal favorite thing is just interacting with folks. We can do ship-to-shore interactions, and that's kind of my favorite part. We get to connect with a lot of our younger audience, so kids usually in summer camps or in little after-school programs that are interested about marine biology. We can actually reach out and talk to them, and it's really nice to try to inspire the next generation of marine biologists and oceanographers.
Interestingly enough, too, geodes are usually spherical, which I guess means we'd have a harder time getting them because we sort of remove those from our pool. Yeah, but rare, I assume, but definitely possible.
So for those of you looking for geodes, circular rocks are your friend. Spherical rocks are your friend.
Can you show me the, oh, never mind, I'll do it here. Can you hit
So I've never done
hit
geode
5
hunting.
on the,
Really?
No. So I'm not I don't know a lot about them.
Other than shiny rock?
I just know it's usually calcite or quartz within them.
we're gonna drop a,
Yeah, the geode we got
Drop some
from the
weights.
Lion Islands was white,
Raj.
so I would imagine,
One of those two minerals,
yeah.
most likely.
Can they come in other colors aside from white?
Oh yeah, of course. Quartz and calcite can be very different, many different colors,
Oh, cool.
especially quartz.
Dropping
Question for the
Oh.
team, do y'all
Dropping
get
the,
seasick? Yes, yes we do.
Dropping the wind chime.
dropping the, yeah,
They are some nice
pepperoni.
chimes, aren't they?
sticks.
Pepperoni sticks.
I think you could get both calcite and quartz too down here. I think they're both saturated. From they could precipitate out of the water column, but and that's how you would form the geode, I think.
Ready for one more step this direction?
Sure.
Okay. Bridge nav. Same step, please. 40300. Thank you.
We have a question for Steve. What time do you expect to have your next dive after this one?
TBD. Yeah, we imagine probably after we recover, we'll have a few hours of sample processing time, and maybe we're not sure yet, but we can probably, there's another guyot within about 4 or 5 hours steam, or it depends on the weather conditions. We might pick another dive site. So we'll determine that over the next few hours.
Back to heading 270.
Roger, roger.
Steve, another question for you. When corals retract their polyps without any disturbance, does that mean that the corals are not actively eating in that moment?
Yeah, I think that's a fair assumption. Was it a fish on the rock?
Yeah,
Here.
Ooh.
it's a nice rock too.
Do a quick
Another
swim on the
tusk eel.
fish.
Yep. Let's see what we got.
They're so tiny.
These deeper fishes, I'm at a loss. So we'll try to get good imagery for our shoreside science folks.
Yeah.
Hopefully get a decent ID.
Oops. Crashing in orbit into the rock there.
Pretty flaky stuff.
Yeah. Now I'm smearing the orbit on the rock.
Fish has had the most exciting moment of its life.
Thin crust.
How about exciting?
We gave that fish an existential crisis in that moment.
Probably burned more energy than he has in the last 3 years. He'll be hungry now.
His friends are never going to believe it.
Yeah, crust isn't, ferromanganese crust isn't as prevalent so far on this dive.
Why do you think that is?
Something to do with the ocean current nutrient system. But
There's something down here. Can we take a quick peek at that?
Sure.
Man, you have a good eye, Steve.
Yeah, he does. Okay, zoom in there.
sure. Crinoid?
Yeah, that's what it looks like. Interesting spot for a crinoid, tucked in a crevice.
That's how you're supposed to park on the bumper.
Cool.
Okay.
All right, thanks. It's very flaky.
Yeah.
It's kind of, I wonder what that would tell us. I would be very curious to see what's on the inside of some of these rocks then.
Stay tuned.
Some of the stuff that's over in this deeper section, it looks pretty altered from, you know, the exposed parts we've seen. And what's behind the flaky crust.
Then really quickly going to answer that question in chat. Are you guys on a guyot right now? No, we're actually in a ridge at the moment.
Yeah, this ridge, think it might be uplifted, where all the other seamounts we've been to, it
Make a
seems
note
more
in the log
volcanic
if
in
you
origin.
want that we drop
Bridge
the
nav.
weights if you haven't.
Roger.
So what's the difference between volcanic and uplifted?
Remember to hand it over.
Other than
Can we
one
move
was made by
40
volcanic
meters
action and
to
one just
80,
happened.
please?
Right.
Thank you.
So uplifted would be just crust that formed at the mid-ocean ridge, got lifted by tectonic processes, where the seamounts have some other source besides mid-ocean ridge. And the bathymetry on either side of this ridge, it's deep ocean, and it's sort of isolated, which makes me think fault on one side and faults on the other side. And faults are indications of tectonic stuff occurring.
Oh, is this the ridge Rob was talking about previously?
It is.
Oh, cool.
So did you say bathymetry?
I did say bathymetry.
What does that mean?
It
Topography,
is,
but underwater.
right, precisely.
Learn new thing every day.
I'm learning lots of new things every day on this cruise.
It is, my mind is officially blown. Oh, here's a question for our ROV guys.
All
Once
right.
you recover, is there a minimum amount of time that the ROVs have to be out of the water for maintenance and such?
Yeah, kind of a pat answer for that is it takes us 4 hours to turn these vehicles around. So by the time we get the vehicles secured on deck, the scientists get all their samples removed, we and do our maintenance and post-dive checks, pre-dive checks, and get everything rigged up to deploy again. It's a busy 4 hours.
Think of it like a pit stop crew at a race car track.
That's exactly what it is. It's a pit stop.
Trying to maximize our dive time, but also make sure that they're in good working order.
Usually 2 of the watches are on deck for that, so we have
More hands.
one of the other watches is whoever is 2 hours closest to their van watch is, we also have them on deck. So there's 4 of us.
They're crinoid.
I think so.
Yep. When we finish this move, I think we can then kind of proceed southwest towards waypoint 3.
Yep, we're starting to change direction that way.
Cool.
It's about 3.5 hours to waypoint 3 at this rate.
Awesome. I imagine there'll be stuff to look at along the way. At about 200 meters to the southwest or so from us right now, or kind of, yeah, right about there. There's supposed to be some sort of bench that we're going to want to look at. So we can put in a temporary waypoint.
Sure.
Just to help us guide our track.
Then to answer the question in chat, are the lasers auto-adjusting to always stay 10 centimeters apart according to how far away they are from Herc? How does that work? I believe our lasers are permanently set at 10 centimeters apart, and we just zoom in and out to see how close or how far they are. And it looks bigger based on how far in we're zoomed. I believe that's right.
Yeah, that's correct. They are parallel, and we periodically check them on the deck to make sure they haven't been bumped, especially if we've been in there working around on the camera where they're mounted.
Do they
Zoom
ever
in there
drift?
real quick, Dan?
Sure.
Bridge nav.
What is that? Oh, it's one of
a
our target.
shrimp.
Yeah, nice eye, Dan.
3rd time's a charm.
Yeah.
Wow.
Yeah, your call if you want to go for it.
Can we move
You wanna
40 meters,
hold off on that
260,
one,
please?
Lynette?
Thank you.
Can you hold off on that one? We're gonna try for a shrimp here.
I feel like Captain Ahab
Sorry,
trying
you
to
want
get
to
these
pause?
shrimp.
Yeah.
Okay. Bridge nav. Cancel that. Thank you.
All right, so we're
See if
going
I
to
can go
try to
for,
take a sample of this shrimp. We've been,
fail number 3 coming up here. You want to show us the,
it's been evasive
see
past
what-
few times.
show me the,
This is the one, I can feel it.
What do you want to see?
never mind.
Well, it's certainly smaller. I don't,
Yeah, everyone at home, root for us.
Are you ready?
Ready?
For that viewer in chat's Discord server, here we go.
Zoom out just a bit.
Sure.
So I can see the
Oh,
nozzle there. Okay, it's close enough. Should be able to go tight now. Which way is he going to jump?
Back towards us, I think, was the...
yeah, they usually will jet backwards.
Yeah, but they go like left or right. They don't go straight back to...
Tell me when.
He's getting jumpy.
Oh.
Oh, he knows. he
Free
knows.
jetting. Okay, go for the suction.
Suction go.
It's a game of wits.
Oh, so
Gosh.
So close.
Nice, I like that approach.
Fail number 3.
I think that was closer than our other ones, though.
Still 8 years away.
closer.
They're fast. They sense the water moving, and as soon as they...
We'll get them.
I think I've gotten like 2 of those things over the years.
Don't worry,
Even
Dan, we're going to make
slow
it
ones.
3.
It's almost like you need a cone,
Yeah,
suction cone.
and Barry has a, if you see some of the pictures of their suction sampler, they have a funnel on the end of it. They're also getting a lot of midwater gelatinous kind of things.
Yep.
So they have a funnel on the end of their slurp gun.
So to take our mind
Let me know
off
when
our
you're ready
shrimp,
for a move.
Yeah,
Yeah.
ready, sorry. Yeah.
we have a
Just
question.
wanted to stop
There's
there
shrimp
while
up
we're-
there.
Bridge
yeah,
nav.
our ship.
Shrimp 3,
Lots of
Herc
shrimp here,
0.
actually.
Can we move 40 meters,
Middle left.
260, please?
Yeah, I see him. We gotta
Thank you.
He's
get
good eye.
going.
Yeah, Raj.
How you see that?
Is that a brittle star on the left?
I think that's maybe a very large crinoid.
Wow, that was cool.
Oh, no, I think you're right.
Yeah, yeah, crinoid, I think.
Oh, nice.
Yep.
Here's a question for ROV guys. Is it part of the ROV touching the rocks that's causing this flaking, or is it more just moving over the rocks with a propulsion system?
Not sure what flaking they're referring to there.
I think the ones you saw earlier, Matt, were the brow, the upper part of Hercules you can see in channel 2, touching
3.
some higher rocks and knocking some flakes down. The way we run the thrusters, Herc is neutrally or positively buoyant, sorry, so the thrusters are actually pushing it down and meant to not disturb the rocks on the seafloor, so it gives us a good viz as best as
Zoom in
possible.
there, Dan.
Yep.
Yeah, the brows, we use the brows to park against the rocks, and depending on the
Another bamboo?
consistency of the rocks, it'll
Some kind
cause
of
stuff to
bamboo
rain down
coral.
into the camera
Yeah,
view.
the brittle star.
You
Yeah,
could go a little
you can
tighter
see
there
there
if you want.
with their tentacle, the arms extended, probably capturing particulates that would get caught up in the arms. I think this is a different bamboo coral than the one we saw earlier. This one is different from the one that has the mound polyps. All right.
Okay.
I don't know what I was
Might as well
expecting,
go
but there's a
tight
lot more
one time.
life than I thought for 3,500 meters deep.
Yeah, I kind of expected it to be like an
Same.
ocean equivalent of a desert.
Yeah, it's I do think it is
Coming up a bit.
more than what we expected, but it's interesting in this case because we're still seeing a fair amount of diversity,
Quick zoom on this guy while we're here. If you want.
but the densities are lower. We just have to travel further often to see similar, numbers of species. Crinoid on a bamboo coral there.
So
I
very
think
large crinoid.
that bamboo coral is different from the previous one. This one is more characteristic of the one we've been seeing for the past couple hours.
What an ornament.
Okay.
It's a star at the top. Yeah.
Yep.
It's like a
He's
Christmas
the
tree.
drunken one at the
Yeah.
party with the lampshade. Drunken coral.
There's some questions in chat asking of how to get in touch with our ship-to-shore interactions. I believe you can find it on our website. If you can contact, I'm not entirely sure who, I believe it's Megan. And you can go to our education tab, and there is a way where you can try to set up an interaction with our ship-to-shore. Yes, if you go to our education tab, you can scroll down to our live ship-to-shore interaction, and then you can click request a connection, and hopefully we can set one up for you.
Bridge nav. Same step, please. 40260. Thank you.
Here's another question regarding the brittle star we saw just a little bit ago. Is the brittle star on the coral a symbiotic relationship or a parasitic one?
Well, symbioses can be positive or negative for one or both parties involved. I don't think it's directly parasitic. It's probably commensal, meaning one animal is getting a benefit while the other is relatively undisturbed. So in that scenario, the crinoid is probably benefiting, whereas the coral probably doesn't have much of a negative or a positive from the presence of the brittle star, or crinoid.
So then, following that up, someone in chat wants to know, what's the funniest or strangest thing you've seen attached to a coral?
Strangest thing, I mean, I've seen lots of marine debris caught up in corals before. Sometimes fishing line and things like that. That's pretty strange, but in terms of biologically, I think some of the funniest instances are where you have gelatinous zooplankton that just gets like smashed into a coral colony, and it just looks like going back to cartoons, like Wile E. Coyote smashes into the face of a cliff or something,
Oh
like
my
just
gosh.
like smashed all over the colony. We see that a lot with Pelagothuria, the pelagic holothurian, or sometimes pyrosomes. And I would imagine the
Another
corals
shrimp.
probably experience a pretty windfall food from that instance.
Another shrimp here, folks.
Oh yeah, how'd the last one go? Did we
Well,
try?
We did. It didn't go so well.
Oh.
0 for 3
It
so
was
far.
really close.
It was the closest one.
You know, I think this is a learning experience, and we've gained wisdom.
As Megan would say, if you get this one, you're batting 250. So honestly, that's still pretty good.
It's in baseball. That's really good.
I'm gonna hold position.
It's not really good, but it's good enough.
I'm gonna say it's really good.
Yeah, better.
All right, fourth time.
Bridge now.
Sounds like baseball is a sport of just like tempering your expectations.
It is, yeah.
Can we hold
Hitting
position
a ball
here,
is
please?
very
Never thought
hard.
about that very much.
It's
Thank
just
you.
like slurping shrimp.
Manage expectations.
That person in chat that lost $50 to their spouse, now's your redemption. No pressure.
It's about
Betting on
$50 on
wow, shrimp hunting.
Betting on
No
shrimp.
pressure.
Sorry, not
No
hunting,
pressure.
collecting.
Please don't do that. It's not healthy.
Read EV Nautilus do not encourage this action.
Okay, go for it. Suction on.
Yep.
Seemed like he started to get sucked, but he's just too powerful.
Yeah. You almost need like a double, you know, hose spout, like one primary hose and then one secondary. You can swap between
cucumber.
the two.
Oh,
Yeah,
wow.
That's a cool looking one.
Zoom on this guy while we're here.
Yep. Yeah, I think this is a type of sea pig. I think it's an elpidiid. Has those legs, you know, modified tube feet. But I don't. Probably elpidiidae. I think that's the best we're gonna do.
Might have just blown it out, but
Yeah.
oh. Yeah, I did. Might get a quick zoom before the dust cloud gets there.
The mouth parts are on the left-hand side
Okay.
there.
Yeah.
To answer that question in the chat, I see your DO data is presented in universal mole
for a move,
mole
Dan?
per liter.
Yeah, ready.
Micro.
Thanks.
What is
Bridge
the
now.
conversion between the scale and the more typically used mg per liter? So the conversion rate for that is you multiply the number by 0.0.
Can we move 40 meters 250, please?
and then you should get it from
Thank
moles
you.
per liter to milligrams per liter.
It's a little bit more complex than that.
Is it?
Yeah, because the relationship is not linear between micromoles per liter and milligrams per liter. There may be some like rough conversions, but most of the time they don't account for changes in pressure or solubility, which could affect oxygen at this depth. Oxygen concentration. So, yeah, I would not say it's a strictly linear conversion, but it's a complicated bit of information. You need to include density in there as well.
So to the person I just said that to, disregard that. Another question for Steve. How tall do bamboo corals usually get?
Another shrimp, top right. Well, unless otherwise acted upon somehow, they would presumably grow for
Do a quick
forever.
zoom there,
You know, I don't know what the typical age or longevity is, but I'm imagining
Full
it's
zoom.
quite old. Yeah, there's the pile of we call phytodetritus there, or
What's this? Kind of a lot of accumulation there.
that's like a smorgasbord for a cucumber or something.
Okay.
Where's that from?
Shrimp. There it is. Oh no.
Yeah, maybe that's why
mortal
we have
enemy.
so many of these shrimp, is that they're eating all this food fall.
So when we do catch the shrimp eventually, what information do we hope to gain?
The shrimp?
Yes.
So there's some open question about the species level identification of this Nematocarcinus shrimp, and there are some experts who are very interested in taxonomy of these critters. It's poorly known, especially at these deeper depths. So this would help support that biodiversity inventory for this region.
All right, Captain Ahab, here we go.
Spastic manipulator.
Yeah.
I was gonna say we're gonna go with the hit it on the head with a rock
I thought
plan.
I'd stun him first.
Are we desperate?
Okay. So, oh, he's gonna
Dire
run away
measures.
before I can get there. I think I'll just leave this thing hooked up to the dredge because we're spending more time chasing shrimp than anything else.
Have we discovered new plants or animals? We're not entirely sure yet, but I'm sure we will hopefully be able to find something if we get our... Okay, so how it basically works is we get our data and samples from the ocean floor, and we put it into our depository, and then we send that depository off to our onshore labs, and then we can see if we discovered a new species or new animal, something like that. I believe that's right. Is that right, Steve?
Yes. What was the full question? I didn't hear the question.
Oh, I was just how we discover. How do we know if we discovered new plants or animals? We send them to shore, and then from there our data, our scientists on there can decipher if we discovered a new plant or species.
Yeah, it's a really kind of community-driven approach. We take input from scientists ashore. Scientists provide input to the cruise plan as well prior to coming out here about requests for specimens, for example.
You can't put
that
the rail
might
cam back
provide,
up
that might support our
Roger.
objectives for the exploration. But in addition, after the samples end up,
Move 40 meters 245, please.
after the samples
Thank
end up
you.
at a common repository, anyone can collect them with scientific interests and conduct whatever tests, analyses they're interested in on them. A lot of biodiversity collections are, they're housed for long term, so they can be, if they're not used immediately, perhaps they can be used sometimes decades, or even maybe these samples will be used for centuries.
That crap column still at 3.
If they're preserved, they presumably have very long-lasting potential to be important collections.
What's the longest sample that you've seen recorded?
The oldest sample I've used in my research was from the 1880s,
Wow.
late 1880s.
And why did you have to go back that far? Was it a rare sample?
Yeah, I mean, even though the pace of ocean exploration has picked up a little bit, back in the day, dredging expeditions were conducted, and a lot of animals and rocks and things were brought up.
Zoom in a bit there, dude.
And a lot of them were housed in museum collections. Nice sea cucumber.
Its gut is so long.
Yeah, this
getting
intestinal
a lot
tract's
of food.
pretty large.
A lot of species were described initially from those collections, but
Right.
we know, we're using different tools these days than dredges and nets, and so we can see things in situ or in their own environment, and that helps us have a better understanding of some of the life history, traits, and characteristics of these animals. And oftentimes we can get a whole specimen rather than a fragment, perhaps. So referencing the older material is important because those are where the original descriptions of these species, and we need to compare, according to the laws of taxonomy and nomenclature, we have to compare with the original, what we call type specimens in some cases if we're doing taxonomic work, to make sure that we're, the animal that we're looking at is similar or different, perhaps, if we're trying to describe a new species.
Understood.
Nice. And then really quickly to answer that question in chat, because I think I can, why is the sea snow in such large pieces here compared to anywhere else?
Oh yeah, another cuskeel.
We believe we're actually just
Good
on top of like a density area. Oh, look at that.
Oh yeah, cuskeel.
Nice. Yeah, that's
I think.
likely a cuskeel.
Is that because of its facial features, or?
Yeah, fin structure too.
That's scarring
Yep.
on its side.
That's common for us.
This
And
one
that
is going to be cooperative, yeah.
That white thing on its side, is that like a... Oh, never mind, it has it on both sides. I was going to say it was a parasite.
It might be a scar from a predation event, like an attack.
So symmetrical on either side.
Yeah.
Yeah, I'm wondering if it was like a synaphobranchid eel came up from the top. It kind of looks like that triangular eel-shaped bite mark.
Bite.
Whoa. Eels eat other eels?
Yeah, I mean, so this the cuskeels I don't believe are true eels. Synaphobranchids, I don't believe they're true eels either, but I have to double check on that.
Gotcha.
Then again,
Ooh, a
why
shrimp.
is sea snow such a large pieces compared to everyone else? We believe we're on top of a density layer, so a bunch of all of that
Oh, they are eels.
layer that's above us sinks down
But
to the ocean
cuskeels
floor
are
in much
different.
higher quantities.
I thought it was the deeper you go, the more they stick together. I forgot the term.
Yeah, both, yeah. It's might be a combination of factors.
Gotcha.
Yeah.
Another question: have you ever found gold on any of these expeditions?
Every
No.
single one.
Yeah.
Biology gold,
Scientific
rock
gold.
Yeah.
Exactly.
shrimp gold. Wow, we're still looking for shrimp gold.
And then those cuskeels that we just saw, do we know how big they get?
They can get pretty big. Yeah, I don't know. I don't have an exact size for you. That's a typical size one, though.
Do you want to pause, Dan?
No, I got
Okay.
that thing all set up here. I'm just
Okay.
going for quick.
The shrimp saga.
The shrimp saga.
Yes. Why do I have the thing all set up?
To answer that
These
question,
are,
Chad.
like really ninja shrimp.
Ninja shrimp.
I'm ready.
They have that
They have an
spidey
escape
sense.
for everything.
I hope this makes into the highlight
Cheeky
reel.
buggers.
They have that spidey
Try
sense.
something a little different. Turn the suction on.
Suction's on. You want to go 90%,
Go
100%?
all the way out. Yeah.
Oh yeah.
Oh.
Oh!
There he goes. Saw him go down the
Not in there
tube.
yet.
Dan.
Finally.
Nicely done.
Nice. Nice.
Fifth time.
That's not in there yet.
Saw him go down the
Turn
tube.
Turn the suction down a little bit.
Suction coming down.
Oh,
there
there
it
it
is.
is.
There
Wow,
it is.
yep.
All right, 0%.
Uh,
Well
Welcome
done.
to your new home.
Great.
And there you
You'll
go,
have
that
to,
guy's Discord server. We finally caught the shrimp.
You can zoom out for us, Dan.
You're at the
You'll have to
200
switch
average
jars
now.
so we don't
All right,
get
so
out.
that'll be useful for taxonomy.
What is that slurp jar for?
I believe that was 4, yeah. Sorry, let me double check.
Nice.
Did you
Thank you.
switch the jars, did you?
Yeah.
Rotate.
All right,
the
I'm
jar.
gonna rotate.
Looks
And
a
then
little
Dan,
tired.
another question for you, Delta Dan specifically. Do pilots ever get sore necks at the end of a 4-hour shift?
Sorry, get what?
Do pilots ever get a sore neck after their 4-hour shifts?
Like at the beginning of the shift. Yeah.
It's always sore.
Oh, don't forget to
All
log the
kinds of
sample and
Contortion.
was the sample logged
Bridge
in
now.
C-log?
I usually do it on the paper first, and then
In the what?
on
The first?
on paper and then C-log, yeah.
Okay.
Can we move
And that's
40
okay.
meters 225, please? Thank you.
As long as as long as they're logged in C-log at the place, because that